Hello. Please report back to your seats for the final presentations of the day. There's 3 or 4. Anyway, come on, back to your seats.
One little announcement while everybody rushes back to your seats. For the bathrooms tonight at Trestle, I just want to clarify how that's working. So when you got a ticket for yourself to be there, it's not a table, it's a person, because we're all going to mix together. But it's a window, so you can go anytime between 5 o'clock and 8 o'clock when the kitchen closes to have dinner. And then it's bat beers, and we're all going to hang out and have fun and drink beers. And you get 15% off because we're special bat people. But the point is, I don't know how we're going to do this, but don't all rush there at the same time. Because they're going to have to try and figure that out in the kitchen. I thought they were going to stage it, but they said, no, just everybody come when you can come. So if some people are going right away, that's great. If other people wait till later, let's try to stagger it if we can a little. Okay.
Pathology and threats. I'd like to invite the lovely Christina Davy to speak to you on her high host specificity of Alphacoronaviruses in Nearctic Insectivorous bats.
Can you hear me okay at the back? Awesome. Okay. Hi, it's really great to be here. I'm really enjoying hearing everybody else's work and thrilled that we were able to make this all work. Thank you to the organizing committee and big much to Shawanaga and especially to Patsy and to Fatima and to Erin for starting us off in a good way this morning. I'm very, very grateful. And of course we are doing our work and the bats that I'll talk about today are living as are we in the Ottawa area, so we are in the traditional and unceded territories of the Algonquin Nation. And the bats that we can work with there are still there because of stewardship for those populations since time immemorial that of course continues into the present day. And so I'm very grateful for that, and I think that it is our responsibility to contribute to that ongoing work through the work that we're doing with bats. So I'm going to talk to you today about viruses in bats. And the particular study I want to focus on has this mouthful of a title, but this is a bat meeting, so this is actually a bat talk. So other than JP and Heather, can I see a show of hands? Who here feels like they are comfortable with the concept of partial RDRP sequencing? Okay, so that's fine. I had to put some jargon in because I'm telling you what we actually did. But when you see the jargon, I promise I will make it make sense to a bat team. Please just stick with me, okay? Don't do that thing where you zone out to the front of your gosh, this is that kind of, don't do that. Okay. So the reason that I'm interested in bat viruses generally is because I'm interested in general in the way that everything is connected, and of course we know, and people have known since time immemorial, that everything is connected, right? The entire field of ecology is founded on this idea. And so I'm interested in understanding bat viruses as part of this big diversity of life, and part of this big cycle of life, and understanding those pathogen interactions. I think some of you know that I did a bunch of work on interactions between the fungus that causes white nose syndrome and bat hosts. And the work that we're now doing on coronaviruses is kind of follow up on that general idea. So, I'm going to skip through this pretty quickly because Burton and others have done a great job. Thank you, priming everybody. You all know about coronaviruses. Important pieces of the coronavirus for the purposes of this presentation are the spike protein, which is on the outside and is involved in the binding of the virus to the host cell, and also, of course, the RNA inside. So this is generally a large family of single-stranded positive sense RNA viruses, and they share this similar characteristic structure. You've all seen this all over the news, right? And they're classified in 4 genera. So we've got alpha, beta, gamma, and delta coronaviruses. And the ones that we're going to focus beta coronaviruses, which are that other blue plate up there. That's the group that SARS-CoV-2 belongs to, and also MERS and the first SARS. But we're looking now at the alphacoronaviruses because those are the ones that are present in our native bats here. So these are our native bat coronaviruses in this area. And you know that coronaviruses can be spread among a whole number of different susceptible hosts. And when a coronavirus jumps from one host to another, from one individual to another, that's just transmission, right? But if it's jumping from one species to another, that's what we call spillover, right? And you can have spillover in multiple different directions. And spillover is going to occur at a point where the spike protein mutates in such a way that it's able to suddenly infect a different type of host cell. That's kind of the general idea. But we don't know a whole lot about coronaviruses in Canadian bats. We know there are coronaviruses in most wildlife out there. If you look hard enough, you will find coronaviruses. In some groups, if you look hard enough, you will find many coronaviruses, right? Berkeley gave us a great example of that. But in Canadian bats, in this study, you can see that our area is really underrepresented for this discovery work. And at the time that this study was published, there were really only 2 studies in Canada. And one of those, and the reason that this is problematic also is that we also don't have a lot of genetic data on the coronaviruses in Canada, in bats and other wildlife. And we have really, it's better in other parts of the world, but it's still pretty limited, as Burton was explaining. So when we do find a new coronavirus, we need to get as close to a whole genome as possible so that we can understand how it's related to the other coronaviruses, we can understand which other species it's likely to be able to infect, and we can also understand how it has evolved and how it's likely to end up where it is today. Whoops. Okay. So in 2017, this study was published. It was led by Dr. Sonu Saludi, who was part of his PhD work in Dr. Vikram Misra's lab at the University of Saskatchewan. And it involved this big team that I was really lucky to be a part of. And Sonu led this description of a new coronavirus, or previously undescribed coronavirus, in little brown bats. Fine. As far as we could tell, this didn't cause any signs of disease. So that's fine, and Burton's already given me the rundown on how bats have these amazing antiviral responses, and so they often don't show signs of infection, even though they're carrying the virus. And then we did a follow-up study where we found that bats that were carrying that coronavirus that were also infected with white nose syndrome ended up with more severe clinical signs of white nose syndrome. And the immune response that they upregulated to fight the fungus downregulated their antiviral response, so they were suddenly shedding more coronavirus. So that was also kind of cool and got me all excited with the idea of co-infections. But of course, these are bats that were sampled in the prairies, which is not where I usually work. And so to try and add more of a viral bat interactions angle to our research program based out of Ottawa, we wanted to understand what's available around us. And so the study I'm going to share now is sort of a first step in setting up more interesting later studies of bats and coronaviruses. So you can access this paper using the QR code if you're interested, but it's a big team effort that went all the way from me and my team collecting actual samples through to Samira Mubareka's lab at Sunnybrook Health Sciences, leading the processing of the samples and trying to understand what was in them, and then a bunch of collaborators from other institutions also helping us to better understand this virus, the 2 viruses that we had found. So I just want to give a huge shout out to this big team because the collaborators were all amazing. I am going to focus on the pieces of this paper that are probably of interest to you as bat people. So if you want to read all the other molecular details, please feel free. But I'm going to try and keep this high level so that I can get the exciting parts across to you. So our workflow was pretty simple. This is the part that my team was involved in. So we collected oral swabs at 5 sites in eastern Ontario. We collected them, some of those in the late summer, and then some of them during hibernation. And we collected them from all 5 species that hibernate in Ontario, right? So we've got big brown bat, little brown bat, tricolor bat, northern myotis, and eastern small-footed bat. The coolest ones. And these are all bats that were given oral swabs while they were being handled for other work. So some of the other studies that people are going to present, those bats also got a little, COVID swab in their mouth very, very gently. It's obviously not going up their nose because it's too small. So very gently swabbed in the mouth and try and get into those cheek pockets.
and then that gets put in a tube and sent over to the biosafety 2 lab at Sunnybrook. Thank you. I just screwed that up. Okay. So basically, the next step was that we had to detect whether there were coronaviruses in the samples at all. And then we identified the coronaviruses that we found. And then we were able to sequence the whole genome of coronaviruses that we found where there was enough material remaining. And so going through the results fairly quickly here, we found that there were coronaviruses in the bats. Not shocking, but cool. So we actually found coronaviruses were present in in samples from 4 of the 5 species we sampled. And we were able to actually get enough material to sequence most of the genome for 2 of those species, so for little and big brown bats. And we found that, I'm just going to stick to the high level here, the 2 detected coronaviruses were both closely related to, but different than previous described strains that were also from those 2 species. So we were able to construct a whole genome phylogeny of these viruses, and you see the brackets around the little brown bat coronavirus up at the top, and then the big brown bat coronavirus at the bottom. And the takeaway from this is that these are nested in this big tangled tree with viruses from a whole bunch of different hosts, some of which are not bats. The light green there is our North American bats, the dark green is Southeast Asian samples, and it's all mixed up, which is telling us that the virus has been jumping back and forth, maybe not often, but fairly consistently over evolutionary time, right? It's constantly being passed back and forth. We don't know what the direction of those spillovers was, but we know that it's constantly happening. We also found this amazing diversity in the spike gene. That's the gene that actually directs infection. So there's a whole lot of mutation going on in that gene, and we're going to dig into that a little bit more. And then, I know this is a terrible slide, I'm not going to bore you with it. This is amazing work that Pavel and Arkadev did, and this is the too long, don't read. Ready? They found that the viral accessory protein that promotes viral reproduction that we found in these viruses, when it's tested in a lab with human cells in a petri dish, so not with an actual human, it is able to inhibit that first line of defense against viruses that mammals mount. And that means that there's the potential that it could actually infect humans. So, in conclusion, we found these 2 coronaviruses. We have a near complete whole genome from them, and they were collected from non-harmful samples. We didn't kill any bats to do this work. We didn't even have to keep them for more than about an hour. And they flew away, and they were just fine, and we were still able to do this surveillance. So it gave us some preliminary insights, and don't go lick your bats. Because I don't think this is a high spillover risk, but it can infect human cells, give it a chance, so don't give it a chance. So just very quickly before I wrap up, Laura, can you wave at everybody? Laura is continuing this work trying to get into the actually ecologically interesting questions that we could answer with this system now that we have it established. And so if you're in a position to collect mono samples, she would love to talk to you. Thank you so much. I'm sorry I went over time.
Thank you so much, Christina. We have time for only 1 question. So who's going to be the lucky person? Okay.
Are you able to get good viral samples from carcasses or from samples that don't require handling live bats?
So the question was whether we can get good samples from carcasses or from other ways to get samples that don't require handling live bats. Yeah, thanks, Phil. Okay, these were swabs, oral swabs, and the detection is actually fairly low. So this is a very conservative estimate of what's there. It's not the best way to detect coronaviruses. Coronaviruses ideally like to hang out in the small intestine, and so the guano samples that Laura is using will give us a better probability of detection than the oral swabs. And the absolute gold standard way to find them is to take the bat apart and use the carcasses. And there's a really cool study that's just come out that did that, and they have some partial sequences from, and they got a much higher level of hits. But when you're using carcasses, they're probably sick. So you can't really relate that to what's going on the landscape. And you can't use it to figure out what the background